So this will be the first part, the isolation technique lab. We're not going to do the entire thing, but I am recording the streak plate now. And as you may remember, the purpose of the streak plate is to isolate bacteria from mixed culture, and I have absolutely glorious mixed culture right over here. I'm trying to find a way to show it to you better. I think at this angle it's kind of okay. So this is a mixed culture. This particular colony here looks very much like what's the word? Fried eggs. Okay. So I will try and isolate this glorious orange microorganism. Okay, I don't know if you can see that. Right here. Right here. It's very orangey. So I'm going to try and isolate. And what I do, I have sterilized loop. I touch this microorganism. I don't really care if it comes out a bit contaminated, okay, with other microbes. And I produce, okay, and I produce a streak. It looks similar to transmissibility lab, but it's nevertheless a little different. So once I produce my first streak, I thoroughly, very thoroughly sterilize the wire loop. I incinerate it well. I make sure that all organic material that was there, all infectious, all live culturable cells are completely destroyed by the fire. Again, it's a little bit harder because of the air movement. But let's say yes, it's done, so we'll let it cool down. We'll let the wire loop cool down. You may often count to 20, 30. It's about the right time for the wire loop to cool down. So now look what I do. I take a swipe to get some of that material, and I start to distribute this material as a second streak. So I essentially diluted my bacteria. I sterilize my wire loop one more time. I let my loop to cool down. And I produce my third streak. So again, oh sorry. So again, make a swipe and I start distributing. Nice and thorough. So that's my third streak. I sterilize the loop. That's the part that I hate the most. Sterilization. And once we're going to be done with that, I'm going to produce fourth and final streak by carefully swiping and distributing my bacteria over some area. Okay. Looks like I did a decent job. Our only overlays where they're supposed to be. So that's our isolation. So what I expect to see, hopefully, in a few days, I expect to see individual colonies, and I will demonstrate them to you, individual colonies of these microorganisms. It probably will be mixed culture, but I will be able to separate species.